Hi everyone, I'm Shichen. Today I will present my final project. The title of my project is Decoding Visual Orientation and Frequency from Neural Activity in Mouse Visual Cortex. One of the fundamental questions in neuroscience is how does the brain represent the outside world? A powerful way to approach this is through neural decoding, where we try to predict sensory stimuli or behaviors directly from neural activity. If a model can successfully decode a stimulus from brain signals, it suggests that those signals contain structured, meaningful information about the stimulus. In this project, we use two-photon calcium imaging data recorded from the mouse visual cortex while the animal views drifting grating stimuli. Our goal is to decode two key features of these visual stimuli, their orientation and temporal frequency. We build deep learning models for this neuron decoding task. If decoding performance is significantly above chance, this will suggest that the recorded neural activity encodes meaningful information about these visual features. So here is a visualization of how neural decoding works. The dataset used in this project is Allen Brain Observatory Visual Coding two-photon imaging data released by the Allen Institute for Brain Science. This dataset contains large-scale in vivo recordings of neuron activity from the mouse visual cortex as the animal passively views a fixed set of visual stimuli. The recordings were collected using two-photon calcium imaging, which allows us to monitor the activity of hundreds of neurons simultaneously with high spatial and temporal resolution. The dataset spans multiple visual cortical areas, various imaging depths, and a range of genetically defined neuron types. In each experiment, an awake head-fixed mouse passively views a series of visual stimuli displayed on a screen. These stimuli include a wide range of content, from drifting gratings and static gratings to natural images, natural movies, and locally sparse noise patterns. The experiment is divided into three separate imaging sessions, labeled A, B, and C, each lasting about 1 hour. Session A presents drifting gratings and natural images. Session B includes static gratings on natural images and natural movies. Session C shows natural movies and sparse noise. To measure neural activity, this dataset uses a technique called two-photon calcium imaging. The mice are genetically modified to express GCA-MP6, a fluorescent protein that binds to calcium. When a neuron spikes, calcium enters the cell and GCA-MP6 produces a strong fluorescent signal. These signals are recorded over time and summarized as delta F over F, which represents the relative change in fluorescence intensity compared to a baseline level. This delta F over F trace serves as the input to our decoding models, providing a time series that reflects neural activity. We show an example of delta F over F trace from a single neuron during one session. You can see how the peaks in the signal correspond to transient increases in calcium concentration, likely indicating moments of neuron spiking. The output of our decoding model is the orientation or temporal frequency of the drifting grating shown to the mouse. These stimuli consist of sinusoidal patterns that move across the screen. The movement is perpendicular to the grating's orientation, which defines the direction of movement. In total, there are 8 orientations, ranging from 0 degree to 315 degree in 45-degree steps, and 5 temporal frequencies from 1 Hz to 15 Hz. Combining these gives 40 distinct stimulus conditions, each presented for 2 seconds, followed by a 1-second gray screen. These 40 conditions are shown in random order, and each is repeated 15 times during the session. Occasionally, weight trails are included as controls. Each trail is labeled with its corresponding orientation and frequency. These labels are what our model attempts to predict based on the recorded neural activity. To simplify our initial analysis, we selected a single experiment from the dataset. We chose an experiment from the primary visual cortex recorded at 275 microns using mice expressing the CUX2 Cree ERT2 Cree line and the AI93 calcium indicator. We used this experiment to develop and evaluate our deep learning models and later used similar experiments with the same brain area, imaging depth, and mouse line to test how well the models generalize across different experiments. Once we selected our experiment, we prepared the calcium imaging data for model input. We focused on trials with drifting gratings, and for each trial, we extracted a 2-second window of delta f over f activity starting at stimulus onset, equivalent to 60 frames at 30 frames per second. This gave us a 2D array of shape number of cells, 60 per trial. Stacking across all trials produced a 3D input array with shape number of trials, number of cells, 60. Each trial also has a label Y indicating the orientation or temporal frequency encoded as integers. For cross-experiment training, we applied zero padding to standardize the number of cells across experiments. Our baseline is a fully connected network. For each trial, we flatten the input and feed it through 2 dense layers with ReLU activation and dropout. Because the input is flattened, this model treats each trial as a static sample and ignores temporal structure. Our second model uses a gated recurrent unit architecture. Instead of flattening the data, we treat each trial as a sequence of 60 time steps. 2 stacked GRU layers process the sequence, followed a dense layer, and finally a softmax output layer. The recurrent connection allows the model to capture temporal structures. We trained each model for 100 epochs, monitoring validation accuracy after each epoch. The model checkpoint with the highest validation performance was saved and used for final testing. For loss, we use sparse categorical cross-entropy, which works well with multi-class classification with integer encoded labels. After training, we evaluated the best model on the unseen test set and used test accuracy as the primary metric. We also examined the confusion matrix to better understand class-level performance, for example, whether the model confuses similar orientations or frequencies. Our results show that both models decode orientation remarkably well. The FCN reaches about 98% accuracy, and the GRU slightly surpasses it at 99%. When we train across experiments, accuracy drops to roughly 93%, indicating robust but not perfect generalization across mice. On the other hand, decoding temporal frequency is substantially harder. The GRU gets about 87% accuracy with a single experiment and only 65% accuracy when tested across experiments. This indicates that frequency-specific information is weaker and more variable across animal than orientation information. The confusion matrix illustrates that misclassification often occurs between orientations 180 degrees apart, such as 0 degrees versus 180 degrees. These are gratings with the same spatial pattern but opposite drifting direction, which may evoke similar activity in the visual cortex. For frequency decoding, errors tend to occur between adjacent frequencies such as 1 Hz and 2 Hz. This suggests that neural activity patterns for nearby temporal frequency are harder to distinguish. These patterns are also visible in the TSNE plots. For orientation, each class forms a clear and separate cluster. Orientations that are 180 degrees apart, like 0 degree and 180 degree, appear close together, which matches the confusion pattern. For frequency, the clusters are less distinct. Nearby frequencies, like 1 Hz and 2 Hz, often overlap. In conclusion, our results show that orientation can be decoded with near-perfect accuracy, while frequency decoding is moderately accurate. Despite reduced performance across experiments, accuracy remained well above chance. This could suggest that shared neural representations exist across animals. These findings suggest that population-level neural activity in mouse visual cortex can encode structured information about visual stimuli, reflecting key properties of the stimulus feedback.